everybody a chance to rearrange. Looks like we've got a good critical mass here, so we'll get started again. It's a pleasure to introduce the second speaker of today's session in room 3, and that is Dhananjay Bhaskar from Yale University. And you've got his title right there, so I'll just turn it over to him.
Well, thank you for the introduction, and thank you to the organizers for giving me an opportunity to present my work today. You can call me Jay, by the way. I'm a postdoc at Yale and also a data science fellow. And my research really lies at the intersection of mathematical biology and mathematical modeling of biological systems and representation learning, machine learning, and topological data analysis of these systems. And as I think you can already tell from the title of my talk, where I'm going to talk about modeling and topological analysis of multicellular architectures, this is going to be a very applied work. So I apologize in advance. I don't have any new theorems and proofs to share with you today. But having said that, there's a consistent theme throughout my talk today where I'm going to relate the empty space or the interstitial space between cells and tissues to geometry and tissue architecture in a way where I believe I can try and convince you that there are some deep connections between persistent homology and percolation theory. So if nothing else, for the mathematicians in the audience, I hope that my talk would be of interest simply because it might prompt you to investigate and discuss these connections between persistent homology of holes and percolation theory. The talk is organized in 2 parts. So first, I'm going to apply topological data analysis to cancer biology. And so no worries if none of these terms make sense. I'll give you a quick primer on cancer biology. And then I'm going to switch gears slightly and talk about applications to developmental biology. And one thing to keep in mind is that the advantage of working on cancer or working on development is that they're kind of 2 sides of the same coin. In fact, cancer cells tend to hijack developmental programs in order to proliferate and to invade into different areas of our body. Okay, so starting with topology of cancer cells, as I'm sure many of you are already aware, cancer starts at some kind of primary tumor sites. These cells start to divide and grow in an uncontrollable manner. They start to invade into their surrounding environment, and eventually these cells find their way into blood vessels that allows them to circulate throughout our body. And then eventually they are able to exit out of these blood vessels and colonize secondary tumor sites. And so we, from a biological perspective, we have a good way of controlling cancer, especially if we detect it in its early stages where the cancer has not yet metastasized. In other words, the cancer hasn't spread to the blood vessels and hasn't colonized any secondary tumor sites. And the way these cancer cells start to invade and start to find these blood vessels is through a process called epithelial-to-mesenchymal transition. So epithelial cells are cells that form the connective tissue in our body. They have a nice, well-organized shape. They adhere to each other quite strongly, and they form this tissue. As part of this EMT process, these cells lose this nice organization. They become very polarized. They become very migratory. They start invading into their extracellular environment. They start cleaving through all these collagen fibers, and that's how they're able to find their way to a blood vessel. And so from a cancer perspective, it's very important that we learn how this process functions, and we find ways to kind of reverse this process or at least stall this process. And so what this looks like in the lab is on the left, you have confocal microscopy here of ordinary epithelial cells. These are from the breast tissue, and you see that they form these nice compact clusters. And on the right in this animation, which I hope you can see, these cells have been induced to go through this epithelial to mesenchymal transition. They are highly motile. They are proliferating a lot. They're not forming these compact clusters. They continue to divide until they form what's called a confluent monolayer, which is that they take up all the space. And it's important to note that this EMT process is not a binary 0-1 process. You can also have something called partial EMT. And so that's the process in the middle, where these cells are a little bit more motile, a little bit more proliferative. And instead of forming these compact clusters, they end up forming this kind of spanning branching network. So we want to understand this process. And as a means of doing that, what you can do is you can start from these microscopy images. You can build yourself a computational pipeline that takes these cell positions and it segments out individual cells and clusters. And you can use the information about cell shape, cell speed, cluster shape, and cluster composition to build yourself a mathematical model. And so in my talk today, I'm going to use a very simple mathematical model that's calibrated using data from these experiments. In my model, I'm going to represent cells as rigid bodies, which they're not. But to a first order approximation, this kind of works. And so I'm going to use non-inertial mechanics, where R is the position vector for a given cell. We have force P here. This is going to be a random polarization force because cells left to their own devices are going to be moving around in random directions. And the second force here, F, is going to be attraction repulsion force, where we have short range repulsion and longer range attraction. The repulsion is simply to prevent these cells from going on top of each other. And the longer range but finite extent attraction is what allows these cells to come together and form a tissue. And so this attraction and repulsion is controlled by this parameter alpha, which I'm going to tweak. So the larger alpha is, the more attractive these cells are to each other. And so the more the likelihood that they're going to form a cluster. And so what this looks like in practice, and I'm going to play all three simulations here at the same time, is at low adhesion and high polarization, they are going to be very motile. They're also going to divide in these simulations, and they're not going to form any kind of cluster morphology. On the right-hand side, with high adhesion and low random force, they are going to form these compact and stable clusters. And in the middle, we have the formation of this branching network or the spanning network. And so it's no surprise, given that our model was calibrated using experimental data, that qualitatively, at least, you see good agreement here between the simulation results and the experimental results I was showing you previously. The color code here is simply has to do with the number of neighbors that a cell has. We also have periodic boundary conditions on all four sides in these simulations. Okay, so quantitatively, and I'm not going to go through this slide in detail. I refer you to our PNAS publication from 2019. But quantitatively, if we measure cluster size, number of clusters, or the radius of gyration of these clusters, there's excellent agreement between these simulations and experiments. So we have a well-calibrated model. And so now that we have a model, we can start tweaking some of these parameter values. And in particular, I'm going to tweak two parameters. I'm going to vary the magnitude of that random polarization or self-propulsion force, and I'm going to vary my adhesion strength. And so at high adhesion and low random force, you have formation of compact clusters. At intermediate values, you have this kind of network formation. And at high polarization, low adhesion, you have cells that are not forming any stable clusters. They're individually disseminated. And if you actually turn on proliferation, so the previous result was with proliferation turned off. Here in the simulation, cells are allowed to divide. So proliferation is turned on. Then you see two distinct phases, maybe three distinct phases, where you have cluster formation. In this parameter regime, you might have some branching with some isolated clusters, or you might have this branching network that forms. What's difficult here is to figure out where these phase boundaries lie. So these dashed lines that I'm plotting here were just kind of manually determined. And so what we found is that persistent homology of cell positions gives us a very nice way of doing this kind of phase classification in a fully automated and model agnostic manner. And the central idea here is that in dimension 0, persistent homology measures connectedness between cells. Well, that measure is biased by the number of cells you have in your simulation or the number of cells you have in your experiment. And we want to be able to classify these things. We want to be able to identify phases irrespective of how many cells we have. So we disregard dimension 0 and we look at dimension 1 homology. So dimension 1 persistent homology tells us the presence of these loops in our data set. And so loops here are going to be cells that enclose some empty space in the tissue architecture. And so we take these cell positions as input, and we compute a persistence diagram that tells us the distribution of these loops over a range of spatial scales. So this is like classical TDA workflow. And so what you're going to find is that when you have a clustered phenotype, you have very few loops, but you have large loops, because those loops are going to represent the empty space that exists between clusters. On the other hand, when you have individually disseminated cells, you're going to have lots and lots of loops, but they're all going to be tiny loops, and they're going to be transient over time. Because these cells are all moving around, those loops are going to get destroyed quite quickly. And so you have lots of loops, but they're quite small. And so you can leverage this to perform the phase classification. So what we do is we compute the Wasserstein distance between any pair of persistence diagrams that we get from our simulation results. And then we use this simple unsupervised classification method to perform this clustering. And so now once you have these clusters, so here we see this, the elements close to the diagonal have higher Wasserstein values. We can take these clustering results and map them back onto the phase diagram. And so what you get here is that you get a classification where we have the clustered phenotype, we have the branching phenotypes, and you have the individual phenotypes. And so this is the clustering result computed in a completely unsupervised manner, using simulations without proliferation. And if you have simulations with proliferation, then we find these three distinct clusters, and the phase boundary ends up being more or less where we thought it would be just by manually looking at these simulation results. So that's all well and good, but this gives us a lot of power. One thing that we can do in particular is we can run experiments in the lab. And so we can take cell positions of actual cells in an experiment, and we can compute the Wasserstein distance between that experimental cell positions and the simulated cell positions. And that allows us to map individual experiments from the lab onto a phase diagram that we have computed using simulation results. And so then we can start tweaking those experiments. So we can start applying various drug perturbations to these cells at different concentrations, and we can see how that mapping moves around in our simulation phase diagram. And so that is a very powerful technique that allows us to connect simulations and mathematical models to actual experiments, and it allows us to quantitatively figure out how experimental perturbations map onto changes to simulation parameters. Another thing that's quite nice about using topology is that the system is very robust. So here we performed two different kind of ablation experiments, where on the left, we take these three phenotypes, individual, branching, and clustered, and we just randomly delete some cell positions. And the reason we do this is because in an experiment, you may not be able to detect all cells. Some cells might be dimmer than others, but we still want our classification to be robust. And what you can see here is that our classification in terms of using Wasserstein distances between these phenotypes, we can distinguish these phenotypes up to a large number of deletions. So the system performs as well as you would do in terms of manual classification of these phenotypes. Similarly, if we take a look at these different phenotypes and we start scaling in, so we start like zooming into these simulation results, we can still disambiguate between these different phenotypes up to a scaling factor of around 2.5, which again corresponds roughly to how well a human would perform at this task. Now, in all the results that I showed you previously, I never mentioned units for my adhesion strength and polarization force. And that's going to bug some people in the audience, especially if you're a physicist. So this slide is simply to say that, yes, you can non-dimensionalize this model. And yes, you can compute a phase diagram with non-dimensional axes. Here we use an adhesion-repulsion ratio and a Peclet number that we defined. But for more details on the model and the approach, I would simply refer you to our publication from last year in Soft Matter. And given 5 minutes that I have left, instead of summarizing this part of the talk, I'm going to move on to the next part of the talk. And so the next part of the talk addresses one of the limitations in this approach. And so there's two main limitations. The first one being that the population was homogeneous. So we had a single cell type. In practice, you might have more than one cell type. And the second kind of limitation is that we didn't really account for temporal dynamics. we computed persistent homology using static cell positions of our simulation results. So it would be cool if we could compute also topology over time. And we assume that that would make our results even more robust. So to address the first part, the first limitation, we now jump to developmental biology. And so in development, it's quite well known that if you have two different cell types, and here we are talking about embryonic cells, and you kind of mix them up, then they will over time automatically configure themselves in one of several ways. They can form like a checkerboarding pattern. You can have one cell type get completely engulfed by another cell type, or you can have them as separated clusters. This is something that's been known for a while. So Armstrong already in 1970s showed that if you take two different chick embryo cells and you mix them up, eventually one cell type gets completely engulfed by another cell type. You can run this experiment at home. So if you go like scuba diving and you find two different species of live sponges, bring them back home, put them in a blender, and just let them sit on your desk, you will find that the two species of sponge are going to self-aggregate and reformulate two sponges. Should mention this only works with living sponges, not the sort of sponges you might have in your washroom. So we can simulate this, of course, and it requires just a minor tweak to our model. So we are going to make our adhesion parameter here, which I had previously called alpha. Now I'm calling it gamma. We're going to make this parameter gamma now dependent on the cell type. So it's going to be gamma ij, where I and j tell us which cell we are looking at, which pair of cells we are looking at. So when you simulate this now with these differential adhesions, what you find is as expected, if blue-blue adhesion is high, but blue-orange adhesion is low, orange-orange adhesion is low, then blue cells form clusters, orange cells stay individually disseminated. If blue-blue adhesion is high, orange-orange adhesion is high, but blue-orange adhesion is low, then they both form separate clusters. And then if you go through the full gamut of parameter values, you will see beautiful pattern formation, including the formation of stripes and spots. And these might remind you of some of the patterns you might see, for example, on the skin of a fish. And one can show theoretically that this whole mechanism goes through a Turing bifurcation, which allows for this kind of pattern formation to take place. And so now that we have multiple cell types, the approach we took to classifying these phases is that we converted our persistence diagrams into persistence image representation. And the way we do this is quite straightforward. We simply take our persistence diagram and we drop a Gaussian at each point in that persistence diagram. And then we kind of superimpose all of these Gaussians. The Gaussians are also weighted by distance to the diagonal. And that's simply to observe the fact that features that are far away from the diagonal are more topologically significant. So we are able to take these persistence diagrams that represent the persistent homology of these cell positions and convert that into an image representation. And the reason we want to use images is because from a machine learning perspective, we know how to deal with images of various kinds. So from this point forward, I'll show you results that we obtained using persistence images of dimension 1 persistent homology of these cell positions. So what do these images look like? Well, I'll show you two different examples here. So maybe I'll go through the top example. Here we have two different cell types, blue and orange. And so you see the persistence. This is the dimension 0 persistence image, which is going to be kind of a flat image because all the points in the persistence diagram in dimension 0 all have a start at epsilon equal to 0, and then they die at various different epsilon values. So this results in a single 1D image. For dimension 1, it's going to be a 2D image. So this is the 1D image. That's the 2D image, a contour plot of the 2D image. And basically, you can tell already just by looking at this image that we have 4 blue clusters because we have 3 distinct peaks that occur at high epsilon values. And those 3 peaks correspond to merging events between these clusters in 0-dimensional homology. You can also see that there is a significant loop among the blue clusters. And that's simply the topological loop that connects all of these clusters together. And it encodes the empty space that's between all of these blue clusters. There's also lots and lots of orange loops, but those are not as significant. They occur at lower epsilon values here. So we take these persistence images and we feed them into a representation learning framework that gives us low dimensional representations of these persistence images that we can then classify using unsupervised machine learning. And that allows us to build this 3D phase diagram where in my diagram, I have blue-blue adhesion on one axis, orange-orange adhesion on another axis, and blue-orange adhesion in a third axis. And the pixel color corresponds to different ground truth configurations of these cells. And so with just 1 minute left, I'll show you kind of the classification result, the punchline, which is that if you look at these persistence images and you look at just a 2D representation through the use of a manifold learning algorithm, you find that, first of all, we have very good clustering. So we can separate out all of these phenotypes. The other nice thing we find here is that it has this nice kind of tree-like structure. And this two phenotypes that are adjacent to each other in this representation or in this tree, they are actually adjacent to each other in the phase diagram. So there exists a common phase boundary between those two features. And so it turns out that persistence image representation, and to a slightly lesser degree, the persistence curve representation, is a fantastic way of classifying pattern formation, both for homogeneous cell populations, but also for heterogeneous cell populations. And we do all of this using only dimension 1 homology. So we only look at empty spaces or interstitial spaces between cells. And that, it turns out, is already powerful enough to complete this task. And just as a final plug, one more limitation that I did not address is that we did not consider temporal dynamics. And so it turns out you need a more challenging problem to take advantage of this temporal information. And so we have a paper out in Chaos. I simply point you to this paper where we do incorporate not just spatial persistence homology, but persistence homology over time to solve a more complex inverse problem where that information can be leveraged. And so with that, just want to thank the organizers again for giving me this opportunity, and my advisors at Brown and my collaborators. Thank you.
All right, let's thank Jay for a nice talk. So I can't resist asking, since you're really doing three-dimensional things here. Any thoughts on the potential for two-dimensional homology being useful?
I am so glad that you asked. I have just a couple of extra slides where we do use dimension 2 homology. And the system here is, again, a cancer system where you will see now 3D spheroids. So in these movies, we have one 3D breast tumor spheroid. And what's going on here is that as this kind of breast cancer progresses, there are important lumens within this tissue that are getting destroyed as these cells move out. And so those lumens are very important because they are the ones that end up forming milk ducts in the breast tissue. And so you can look at the organization of these lumens by looking at dimension 2 homology. And what we are doing now is we are looking at generators. So given the topological barcode for dimension 2, we ask what are the cells that lead to the formation of this hole or this void? And then we want the minimal kind of set of cells that lead to the generation of this void. So we leverage these generators to then look at these empty spaces in that tissue. And what you can then do is you can figure out which cells belong to a lumen and how these cells flow from one lumen to another lumen or from one lumen to like the extracellular space. And so you can put together a diagram like this, where you are kind of tracking which cells belong to each lumen and how they're going from one lumen to another or to like some other place in the tissue. And that's extremely important especially in the context of breast tumors.
All right, thanks. Other questions? Go ahead and unmute and jump in if you'd like.
I had a quick question. I really enjoyed this talk. When I saw the clustering that you did in the first project, I was kind of interested if you tried anything more sophisticated than, so you used Wasserstein distance with single linkage, right?
Yes.
Yeah. So did you try, I mean, you can do k-means in at least 2 different ways for persistence homology that I can think of. Did you try anything of that nature?
So in the very first project where I presented, let me see if I can bring up the slide quickly. Okay, so these are all, this is the single linkage of Wasserstein distances. What I also tried was the bottleneck representation, which gives you very similar results. But in terms of the clustering itself, I used hierarchical clustering in almost all the projects that I presented today. The project that I did not get to present on using both temporal and spatial information, in that project, we tried various different unsupervised methods of clustering, including k-means and k-medioids. More or less, I think as long as the representation is strong enough, all clustering methods give you same results. The question is how many clusters you want as an output from the clustering algorithm. And for that, we use things like silhouette score or gap statistic to kind of estimate how many clusters exist in the data. But by and large, most clustering algorithms give similar results.
Cool, thanks.
All right, time maybe for one more quick question if anybody's got it. All right, let's thank our speaker again.
Thank you.